Sample contamination chances are there between the sample collection tubes. Chemistry sample collected after potassium EDTA. The increased level of potassium available in the lavender tube can contaminate the chemistry tube. Hello friends, welcome to TechQuest channel. This is for the medical techs by your tech. In this video, we will discuss about the order of blood sample collection and why it's important. We use vacuuminers for blood sample collection nowadays. Earlier days, we used to collect in sterile syringe and transfer it into a test tube or anticoagulant containing tubes or bottles. Vacuuminer collection methods are better because of its closed system and the volume of sample is collected by vacuum present inside the test tube that makes the exact proportion of anticoagulant to blood ratio. Vacuuminer methods avoid external contamination of samples by microorganisms, but sample contamination chances are there between the sample collection tubes. So the order of draw becomes important to minimize the contamination of anticoagulants present between the tubes. For example, chemistry sample collected after potassium EDTA. The increased level of potassium available in the lavender tube can contaminate the chemistry tube and elevate the potassium level. Same way, the heparinized test tube can contaminate coagulation studies, that is PT, PTT, INR results if citrated tube is collected after heparinized tube. So, we need to make an order of draw to minimize these contaminations. Quality control policies should provide the protocol for order of draw in each lab, and the phlebotomy supervisors and quality control managers periodically check and make sure these orders are strictly followed. Okay, let's see the order of draw. The first is blood culture. Blood cultures are drawn first as we do not want to contaminate with any anticoagulants that can suppress the growth of microorganisms. The blood culture container contains nutrient broth for the growth of microorganisms, ingredients that can suppress the phagocytosis by the blood cells after collections, the reagents that can inhibit complement activity because we don't want the microorganisms to be killed by phagocytosis or complement activity. The nutrient broth present in the container also dilute antibiotic present in the sample if the patient has already taken antibiotic. All the reagents helps the microorganisms viability and multiply for the identification. Next, we have aerobic and anaerobic cultures. Which is first? Aerobic container sample is first because the air present in the needle and the vacuuminer can contaminate anaerobic cultures. So, first is aerobic and then anaerobic. Next tube is pale blue tube used for PT, INR, PTT and other coagulation studies. This tube contains sodium citrate of 3.8% or 3.13 moles. The blood coagulant ratio is 9:1. Since it contains very low level of citrate, the chances of contamination to the next tube is minimal. The next tube is for chemistry studies, SST tube or the red top tube. These tubes contains clot activators and the sample is expected to clot fast so that the serum is separated and analyzed within short time period. Next is golden top gel tube used for biochemistry test. These tubes contains gel that can separate the blood cells and serum after centrifugation. Some of the analytes can be used by the cells even after collection of blood. Keeping cells and the serum or plasma together in the test tube for long time can decrease the analyte level. Example, glycolysis is usage of glucose in the serum or plasma by the cells in the test tube and decrease the glucose level. So the gel tube present between the serum or plasma and the cells minimize the usage of the analyte by the cells. Next tube is heparin containing green top tube. Heparinized samples are used for lactate, ammonia, ionized calcium test. These tubes are placed in ice containers to maintain a low temperature before testing. Next is the lavender top tube containing potassium EDTA, commonly used for hematology test and HbA1c in chemistry lab. Next tube is the pink top tube, contains EDTA used for blood grouping and cross matching in blood bank. The next tube, the last tube, is a gray tube containing fluoride or oxalate used for testing lactate level or ethanol test. The amount of anticoagulant is more compared to other tubes, so that this tube is collected in the last to minimize the contamination to the other tubes. All the anticoagulated tubes must be mixed gently for 10 seconds. No vigorous mixing is needed as these movements can lyse the cells. Sample should be labeled immediately after collection, never labeled before collection, as this can lead to mismatch the sample if sample is not collected for a patient for some reasons, and next patient sample can be added to these labeled tubes. Thanks for watching. Please like, subscribe and write your comments to make more videos. Thank you.